How does an infant's brain map the visual world? Today we'll explore the remarkable process of retinotopic organization in human infants and how their visual system begins making sense of what they see. Today's presentation is by Nicole, Kate, Carson, and Claire.
Vision is the most dominant human sense and begins to develop starting in childhood all the way to early adolescence. Our visual processing originates in the visual cortex, where images from the retina get processed within the 6 layers of the visual cortex. Beginning with V1, known as the primary visual cortex, it sends the basic information to V2, V3, V4, and V5 for more higher order analysis and processing.
This information is then processed and depth, perception, form, and color are all perceived through higher order structures. V2 is the secondary visual cortex and receives information from V1 and passes it along to V2, V3, and V4. V3 plays a role in processing motion and color sensitivity.
V4 is very responsive to color and part of the ventral processing stream. V5 contains cells that are highly sensitive to motion.
The visual cortex is organized topographically, which means that a visual stimulus that's presented at a specific location in the visual field will also activate a corresponding location in the topographic visual map. The early visual cortex, which is the focus of the early retinotopic organization of the visual cortex discussed in the paper, is responsible for encoding low-level features of visual stimuli, such as contrast and orientation.
A retinotopic map consists of eye-centered spatial relationships where adjacent neurons along the cortical surface receive input from adjacent points on the retina surface. Multiple retinotopic maps of the visual cortex suggest that the visual cortex is organized into many areas. In addition to having retinotopic maps, there are also spatial frequency maps that let visual areas respond to different frequencies of stimulation.
Before this research was conducted, animal studies indicated early organization in the primary visual cortex with slower maturation of higher visual areas, suggesting a foundational map for visual processing exists from birth. And human infant research indicated that basic visual abilities emerged shortly after birth, but there was a limited direct evidence of detailed retinotopic mapping in human infants, especially for complex areas beyond V1. Additionally, functional connectivity suggested some indirect findings that basic visual boundaries in infants, but direct retinotopic evidence was missing.
Before birth, our brains begin organizing a structure that connects the eye's signals to the brain in a way that preserves spatial information. In newborns, certain areas in the visual cortex, like V1, start showing some adult-like responses, while other areas, like V2 and extrastriate regions, are still immature. This delayed maturation is important because it allows the brain to adapt to visual experiences over time, which helps build complex visual skills. For human infants, however, we don't fully understand the timeline of this visual development. Most of what we know comes from studies in macaque monkeys, where researchers have observed that the extrastriate cortex, which is responsible for advanced visual processing, develops after simpler visual areas. Human infants likely follow a similar but slower process. We see basic visual behaviors like recognizing orientation at birth, while more complex abilities like contour integration take months or even years to fully develop.
The study aimed to understand how visual organization progresses in infants and specifically how to map out the early structures of the visual cortex, specifically in areas V1 through V4, and how these mature over age. To understand how visual areas in infants' brains are organized, the researchers used a specialized fMRI protocol that works with infants who are awake and actively engaging. This included infants from 5 to 23 months old. They aimed to map out key visual areas, specifically the ventral and dorsal regions of V1, V2, V3, and V4. These are all parts of the visual cortex that process different kinds of visual information. And to do this, they used 2 main techniques: meridian mapping and spatial frequency mapping.
Meridian mapping uses visual stimuli that alternate between horizontal and vertical lines. By seeing how the brain responds to each orientation, the researchers could outline the boundaries between different visual areas such as V1, V2, V3, and V4. This is similar to tracing a map of these regions in the infant's brain. Small animations were shown to encourage fixation. The meridian mapping phase is followed by a rest period of 6 seconds.
Spatial frequency mapping, patterns with high and low spatial frequencies were used, including both finely detailed patterns and less detailed patterns. This helped identify how receptive fields, which are the part of the visual field each neuron responds to, are organized in infants, similar to what's been found in macaque monkeys.
Let's recap the methodologies to make sure we are understanding. First, patterns with high and low frequencies were used to help identify how neurons respond to receptive fields. Then, meridian mapping blocks were used that were positioned horizontally or vertically to identify the difference between visual areas in the brain. Then, there are alterations between 20 seconds of stimuli with 6 seconds of rest, all while being visually stimulated by moving shapes.
To study the organization of infant visual cortex, researchers utilized general linear models to estimate bold responses in 4 distinct stimuli. This allowed them to analyze the visual cortex's response and test for evidence of retinotopic organization. First, it was confirmed that robust activation of the infant cortex across all stimulus conditions. The results showed strong activation. Next, the researchers compared horizontal versus vertical meridians and low versus high spatial frequencies. To visualize these patterns, surface reconstructions were created using IB. The reconstructed surfaces were then inflated to remove cortical folds and then cut and flattened to generate maps for easier visualization of the visual areas. Contrasts between meridians were projected onto flat maps. The researchers traced visual area boundaries based on alternations in sensitivity to the meridians. To verify the presence of gradients of selectively for different meridians across the visual areas, the team traced lines perpendicular to the area boundaries. Ultimately, this provided quantitative evidence of the gradients and supported the existence of distinct visual areas in the infant cortex.
To understand how infants engaged with the visual stimuli, the researchers carefully monitored their gaze during the experiment. This was done by recording the infant's face on video and then manually analyzing where they looked. For the meridian mapping blocks, infants generally looked at the horizontal or vertical meridian throughout each block. However, they didn't always fixate on the center of the screen. Instead, they often looked slightly off-center to the right or the left at the center for horizontal meridians, and above or below the center for vertical ones. In the spatial frequency blocks, where infants saw high and low detail patterns, they also tended to keep their gaze on the screen. Interestingly, they looked off-center more often during the high frequency, so the more detailed blocks, rather than the low frequency ones, suggesting that they might need to focus more precisely on the detailed patterns.
They also found that younger infants had lower quality data compared to older ones, mainly due to more frequent gaze shifts or unusable gaze data. Despite this, the overall results did not show a strong improvement in data quality based on the infant's age within their sample.
To see if infants' visual cortex shows signs of retinotopic organization, the researchers estimated brain responses to 4 types of visual stimuli. First, they confirmed that the visual cortex was strongly activated across all conditions, and they saw distinct patterns when comparing horizontal versus vertical meridians and high versus low spatial frequencies. This suggested that infants' brains respond differently to each type of visual input. A tool called IB was used to create 3D surface reconstructions of each infant's brain. These surfaces were flattened to make clear maps, and they projected the contrast between meridians onto each map. By focusing on where these orientations peaked, they traced out visual area boundaries, like the border between V1 and V2, which they identified using a strong response to the vertical meridian in a specific part of the brain. Remarkably, they could identify these visual areas in 16 out of 17 infants. To verify, they traced lines across boundaries and saw shifts in sensitivity between horizontal and vertical meridians, showing consistent patterns across participants. Even more exciting, 3 infants under 6 months old showed these same patterns, providing strong evidence that this early organization in the visual cortex is present from infancy.
So how does the visual cortex respond to different levels of detail or spatial frequencies? They used spatial frequency tuning as a substitute for traditional mapping. They knew from prior studies that in adults, high-frequency patterns activated areas near the center of vision, while low-frequency patterns activate areas further out in the periphery. VoxOY's sensitivity was analyzed between high and low frequencies using brain imaging data. The findings showed that the infant's brain sensitivity to different spatial frequencies is consistent across infancy, which means that infants, like adults, likely have areas tuned to different spatial frequencies in both central and peripheral vision. A larger response in the periphery to low-frequency patterns was observed, suggesting that even in infancy, the peripheral areas of the brain have relatively larger receptive fields.
Instead of tracking each infant over time, the researchers compared manual tracings of visual areas between infants of similar ages. They tested 2 main things. First, age similarity, showing if infants close in age had similar results, and second, maturity effect. They found a positive correlation suggesting that older infants maps are becoming more like the stable organization seen in adults. They also looked at how well visual landmarks like gyri and sulci aligned with age but found no strong relationship, which suggests that these structures do not change much over infancy. Overall, the increased similarity among older infants was interpreted as a sign that infants' visual cortex is gradually converging toward an adult organization. However, even the youngest infant showed high similarity to adult configurations, suggesting that early brain organization provides a foundation for development. Early on, only visual areas actually change in size as infants grow, which contrasts with the idea that visual cortex develops strictly in a step-by-step hierarchical way. Instead, this shows that much of the visual cortex's basic layout is already in place from an early age.
While there was a small increase in the size of V1 with age, the general layout and boundaries of these areas did not change from 5 to 23 months. This suggests that during infancy, the development of the visual cortex might be more about refining and adjusting the existing structure rather than reorganizing it. The study also observed organized responses in the extrastriate cortex, suggesting that some of the fundamental architectural features like gradients and receptive field size are in place from a young age. This organization provides a foundation that supports specialized visual functions as the brain matures. Visual processing abilities during infancy might not depend on new visual areas forming, but instead on fine-tuning and strengthening the early organization already present in the visual cortex.